Now first we will study about mitosis. Mitosis is the equational division, and the name mitosis was given by Walter Fleming in 1882. Mitosis has four phases: prophase, metaphase, anaphase and telophase. Prophase means before phase, and that is the first phase. Metaphase is the mid phase, ana means back phase, and telo is the end phase. Coming to the prophase, prophase is the first phase where a slow condensation of chromatin fibers occur, and the condensation leads to the formation of chromosome. One chromosome has two sister chromatids, and these two sister chromatids are attached together at a central point that is called as centromere, and they are bonded together by a cohesin protein. This cohesin protein is a member of SMC protein, and this SMC protein is structural maintenance of chromosome. This cohesin is a protein complex, and it has further two subunits. The subunit first has SMC1 and SMC3 ATPase, while the second subunit has SCC1 and SCC3 that connects to the ATPase domain. This cohesin binds and put the two sister chromatids together at the centromere. In prophase, there is the disappearance of nucleolus or nucleoli, that is the main point or structure that is present and involved in ribosomal RNA synthesis. In prophase, there is the disruption of nuclear envelope. Thus, four steps occur in the prophase. First, the chromatin condensation, then sister chromatid attachment and disappearance of nucleoli, and the fourth is the disruption of nuclear envelope. Coming to the second phase, that is metaphase, that is the mid phase. In metaphase stage, there is alignment of chromosomes at metaphase plate. This alignment is called as congregation, and it occurs via microtubules. This microtubule filament formation occurs by centriole. The two centrioles known as the centrosomes are present at the two poles of a cell. The alignment of chromosomes occur by the attachment of microtubules at its plus end via kinetochore, that is a disc shaped structure having NDCAT complex, that is a rod shaped protein, and on the other end they are attached to these centrioles. These centrioles form three types of microtubules. These are polar microtubules, second is the kinetochore microtubule, and third is astral microtubules. The polar microtubules are those microtubules where the microtubular plus end grows opposite to the second pole, but they overlap each other and there is no attachment of chromosome. They just overlap each other and they are called as interpolar microtubule. The second type is kinetochore microtubule, where the microtubule grow from their negative to plus end, and their plus end attaches to the kinetochore of the chromosome, condensed chromosomes, and similarly the second microtubule from the second centriole that is present on the second pole also attaches to the second kinetochore of the chromosome, and thus the alignment of chromosome occurs here. This is called kinetochore microtubule. Third is the astral microtubules, where the microtubules are growing at the periphery of the cell or cortex of the cell, and there is no attachment of chromosomes here. Thus, in metaphase stage, there is only the alignment of condensed chromosome on the metaphase plate, and they are attached via kinetochore and NDCAT complex, and on the other end they are attached to the microtubules. Now the next phase is anaphase. Ana means back phase, that is just after the metaphase or mid phase. At anaphase, the two sister chromatin get separated and they move to two opposite poles. During separation and movement, the tail of this chromosome or the arms of this chromosome is trailing behind. The separation and movement of the sister chromatids depends on different proteins. The movement of sister chromatid to opposite poles depend on the regression or the shortening of the microtubules that are present at the two opposite poles, called as centromeric points. But the two sister chromatids separation at anaphase depend on the presence of separase. Separase is a protein. Initially at metaphase it is binded with securine, but at anaphase it is free and it causes the degradation of cohesine protein complex that is present at the centromere that bind the two sister chromatids together is degraded, and due to degradation of this cohesine protein the two sister chromatids get free. Thus at anaphase the sister chromatids get separated due to the degradation of cohesine and they move to two opposite poles due to shortening of the microtubular proteins. Now the last phase is the telophase, and it is also called as the end phase of mitosis. In telophase 5 sequential events occur, where the first event is that two sister chromatids move to two opposite poles and the regression of microtubules occur here, having the negative and plus end. Second is that nuclear envelope reappears or reforms around the two chromosomes separated at two poles. The third step is that nucleoli again reforms or begin to reform in the nuclear envelope region. Then the decondensation of the chromosome occur and they again form the thread-like structures. Then the cellular organelle start to reappear in the cytoplasmic region of the cell. Thus we can say that telophase is just opposite to prophase stage. Now we know that mitosis include both karyokinesis and cytokinesis. The karyokinesis is the division of nucleus or chromosomes, and cytokinesis is the division of cytoplasmic material of a cell. But this cytokinesis is variable in animals and plants. First we will study about the cytokinesis in animals. In animals, the cytoplasmic division is based on the contractile ring formation. This contractile ring formation is made up of actin filament and myosin type 2. This contractile ring is made by organized bundle of actin filaments and myosin type 2, and the process is called as cleavage furrow formation. This contractile ring structure shows the contraction and the muscle-like sliding. By the contraction or sliding of this actin and myosin filament here, it leads to the division of cytoplasm with the separate nucleus forming two daughter cells, daughter 1, daughter 2 from the mother cell. Is cytokinesis in plants. In plants, cytokinesis occurs by formation of phragmoplast. In plants, they have an outer hard covering known as cell wall, a cell membrane, and then in G2 phase during mitosis process they form an array of cortical microtubules, and this microtubular cortical structure is called as pre-prophase band, and it is present just beneath the plasma membrane or cell cortex region. This cortical structure further forms phragmoplast and it occurs at anaphase stage. This phragmoplast also has some part of actin filament. Later, this phragmoplast grows outward toward the periphery of the plasma membrane and causes the division of this cytoplasm into two halves. Later, this phragmoplast fuses with some Golgi vesicles, and the membrane of this vesicle forms the plasma membrane of the new daughter cells. Further, this phragmoplast fuses with cellulose fibriller. These cellular microstructures are called as cellulose microfibrils, and these cellulose microfibrils fused with phragmoplast and form the outer covering or the cell wall. Thus, by the division via phragmoplast, further the Golgi vesicles of Golgi form the plasma membrane, and cellulose microfibrils fused with this phragmoplast form the cell wall. Thus, all the layers of the plants are formed and the cytoplasmic division of the cells occur. Now, the mitosis part is over, and in next class we will study about meiosis and the mechanism of cell cycle regulatory checkpoints.